Mutation in gene regulation. Mutation. A mutation is a change in a DNA sequence. Mutations can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses. A mutation occurs when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. A mutagen is an agent of substance that can bring about a permanent alteration to the physical composition of a DNA gene such that the genetic message is changed. There are three types of DNA mutations: base substitutions, deletions, and insertions. These base substitution are the simplest type of gene level mutation, and they involve the swapping of one nucleotide for another during DNA replication. With base substitution mutations, only a single nucleotide within a gene sequence is changed, so only one codon is affected. Deletions is a type of mutation involving the loss of genetic materials. It can be small, involving a single missing DNA base pair, or large, involving a piece of a chromosome. A deletion mutation is a mistake in the DNA replication process which removes nucleotides from the genome. A deletion mutation can remove a single nucleotide or entire sequences of nucleotides. Insertions is a type of mutation involving the addition of genetic material. An insertion mutation can be small, involving a single extra DNA base pair, or large, involving a piece of a chromosome. An insertion mutation has occurred if this mutation was in a gene or the part of a DNA sequence that codes for a protein. It could be detrimental and results in the production of a non-functional proteins. Gene regulation. What is the meaning of gene regulation? Gene regulation refers to the mechanism that acts to induce or repress the expression of a gene. Gene regulation is the process of turning genes on and off. During early development, the cells begins to take on specific functions. Gene regulations ensures that the appropriate genes are expressed at the proper times. Gene regulation can also help an organism respond to its environment. There are three broad levels of regulating gene expression, and these are transcriptional control, translational control, and post-translational control. So, transcriptional control is the means by which a cell regulates the conversion of DNA to RNA, thereby orchestrating gene activity. First, transcription is controlled by limiting the amount of mRNA that is produced from a particular gene. The second level control is through post-transcriptional events that regulate the translation of mRNA into proteins. Translational control is an important mechanism by which cells govern cell gene expression, providing a rapid response by the cell without invoking nuclear pathways for mRNA synthesis and transport. Translational regulation refers to the control of the levels of protein synthesized from its mRNA. This regulation is vastly important to the cellular response to stressors, growth cues, and differentiation. And the third is the post-translational control. Refers to the control of the levels of active protein. There are several forms. It is performed either by means of reversible events or by means of irreversible events.